There are two types of cells, prokaryotes and eukaryotes. Prokaryotes divide through binary fission, which is when genetic material is duplicated and cytokinesis happens, making two identical cells. Eukaryotic cells go through mitosis and meiosis. Somatic cells go through mitosis to create two identical daughter cells, while meiosis creates four genetically diverse daughter cells in sex cells.
Interphase is the overall growth period in a cell where a cell will spend 90% of its time. Interphase consists of G1 phase, when the cell grows and develops normally, S phase, where genetic material is duplicated, and G2 phase, where the organelles are copied and nuclear envelope forms to enclose the nucleus. A cell will go through G0 phase when it is not preparing to divide.
Mitosis is the process that occurs in somatic cells for nuclear division. Mitosis conserves chromosome number by allocating replicated chromosomes equally to each of the daughter nuclei. In prophase, the duplicated chromosomes appear as two identical sister chromatids joined to their centromeres. The mitotic spindle begins to form and is composed of its centrosomes and microtubules. In metaphase, chromosomes line up at the metaphase plate at the center of the cell. For each chromosome, kinetochores on the sister chromatids are attached to kinetochore microtubules coming from the asters.
Anaphase is the shortest phase of mitosis. The sister chromatids or daughter chromosomes are pulled apart. The daughter chromosomes begin moving to opposite ends of the cell as kinetochore microtubule shorten. The cell elongates to an oval shape. At the end of anaphase, each end has a complete set of chromosomes. In telophase, the daughter nuclei form in the cell. Nuclear envelope and nucleoli reappear. The spindle microtubule depolymerizes. During cytokinesis, the cytoplasm divides between the two cells. In animal cells, the cleavage furrow pinches the cells in two. In a plant cell, a cell plate forms from vesicles, which turns into a new cell wall.
At the end of mitosis, there are two identical daughter cells with an equal number of chromosomes. Every human cell has 23 pairs of chromosomes. In interphase, prophase, metaphase, and telophase, each cell has 23 pairs. In anaphase, when the sister chromatids are splitting, there are double the amount of chromosomes in the cell. At the end of mitosis, the chromosome number remains the same.
Meiosis is the division of sex cells that produces four genetically diverse daughter cells, which is important for genetic diversity. Meiosis occurs in sex chromosomes, which are the X and Y chromosomes. Meiosis has two cycles. Meiosis 1 separates homologous chromosomes, and meiosis 2 separates sister chromatids.
In prophase 1, chromosomes begin to condense and homologs loosely pair up, aligning their genes. At the chiasmata, crossing over occurs, which is when homologous chromosomes swap corresponding DNA. Centrosomes begin moving to opposite ends of the cell, and the nuclear envelope breaks down. In metaphase 1, pairs of homologous chromosomes line up at the metaphase plate, with one chromosome in each pair facing opposite poles. Both chromatids of one homolog are attached to kinetochore microtubules from each pole.
In anaphase 1, breakdown of proteins allow homologs to separate. The homologs move towards opposite poles pulled by the spindle apparatus. At the beginning of telophase 1, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. Cytokinesis occurs simultaneously with telophase 1. In an animal cell, a cleavage furrow forms. In a plant cell, a cell plate forms.
In prophase 2, a spindle apparatus forms. Chromosomes, each still composed of two chromatids, move toward the metaphase 2 plate. In metaphase 2, the chromosomes are positioned at the metaphase plate, similar to mitosis. Because of crossing over, the chromosomes are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles.
In anaphase 2, breakdown of the proteins holding the sister chromatids together at the centromeres allow the chromatids to separate. The chromatids move towards opposite poles as individual chromosomes. In telophase 2, the nuclei forms and cytokinesis, division of cytoplasm occurs. The meiosis division produces four daughter cells, each with a haploid set of chromosomes. The four daughter cells are genetically different from one another and from the parent cell.
Meiosis produces four genetically diverse haploid daughter cells. The daughter cells eventually turn into gametes. In prophase 1 of meiosis, the sex cell has 46 chromosomes, one chromosome from the father and one chromosome from the mother. After telophase 2 and cytokinesis, each cell has 23 chromosomes. At the end of meiosis, the chromosome number is reduced by half.
Mitosis is a cell reproducing asexually, while meiosis is a cell reproducing sexually. Mitosis occurs in all organisms. Meiosis only occurs in organisms that reproduce sexually. The daughter cells produced in mitosis are identical. The daughter cells produced in meiosis are genetically different. Crossing over only occurs in meiosis. There is one cell division in mitosis and two cell divisions in meiosis. Mitosis results in 2 diploid cells, and meiosis results in 4 haploid cells. Meiosis creates sex cells, and mitosis creates every cell but sex cells.